Psychiatric disorders such as depression and anxiety are strongly associated with an increase in mortality rate, a decrease in quality of life, and notably reduced cognitive ability. This summer I conducted research on a Manitoban cohort who had conditions called an IMID, an immune-mediated inflammatory disease, which includes disease such as rheumatoid arthritis, multiple sclerosis, and inflammatory bowel disease. These IMIDs are strongly associated with depression and anxiety, so we wanted to validate the transferability between IMIDs and reduce cognitive ability using something called polygenic scores, while also comparing the level of reduced cognitive ability by the three immune-mediated inflammatory diseases. We want to predict reduced cognitive ability, or IQ, in IMID patients with polygenic scores. Now, these polygenic scores takes in consideration multiple genetic variants, either looking at multiple genes that are associated with and influence cognitive ability, or even looking at minute differences down to the nucleotide within a particular gene. We find this data from genome-wide association studies, and from there we can generate polygenic scores using various regression models using the R programming language to generate a score that represents, in this case, the cognitive ability of a participant. To assess the level of cognitive ability, we use the Wechsler test of adult reading, a test that assesses vocabulary levels using one-syllable words with an irregular pronunciation. We also collected data on the cohort to find characteristics such as age, sex, race, marital status, income, and education level. We also identified patients with an IMID that also had a pre-existing depressive and/or anxiety disorder. The results were that the beta estimates in our regression model showed that an increase in IQ polygenic score was associated with a lower reading test score, an inverse relationship between the two. In other words, people at a high risk for reduced cognitive ability had a lower test score overall. However, none of the regression models across all measurements yielded statistically significant data. The limitations of this project were two main things. One factor was outdated standards with the Wechsler test assessments, which forced us to use raw scores in this project instead of a standardized score. A second factor was a control group. In order for us to get a clear look into interpreting these results, we must update the genotype for these healthy controls to compare the IMID cohort with. This project was done to provide insight into whether the cumulative genetic burden for IQ are associated with cognitive abilities in the Manitoban cohort. This project is still a work in progress, and there's a lot to be done. Data should be investigated further to be able to translate this knowledge into a clinical setting. Thank you.